There are two kinds of phosphorylation. As I said, phosphorylation is the phosphate group add to nucleotide, okay, and addition of phosphate group into ADP or AMP, okay, so we call it as phosphorylation. There are two types. One happens in the cytoplasm, another one happens in the inner folded membrane of the mitochondria. So substrate link phosphorylation, okay, phosphate link, phosphate is directly transferred from the substrate to ATP to form ATP. So energy is required, okay, this is the main point, required at the beginning, occur in the cytoplasm and also in the matrix of the mitochondria. Okay, this one we will further discuss in the matrix. So where is the matrix? Matrix is inside. Okay. Now, for only small amount of ATP synthesized during aerobic respiration. Okay, number 5, produce 4 to 6 ATP per glucose molecule takes place in glycolysis. Okay, maybe you are confused with the aerobic here. So the third part we will talk about in this aerobic respiration in details. Chemiosmosis involve a proton. Proton means hydrogen atom losing one electron. So it is a hydrogen ion, or we call it as proton gradient. Proton gradient across a membrane. So it is something like this. Okay, so the proton, the hydrogen ion pass through the inner membrane. Okay, so from high concentration to low concentration, or from low concentration to high concentration, we will further discuss chemiosmosis. Okay, takes place across the inner membrane of the mitochondria. This is the one, okay, and the thylakoid membrane of chloroplast, thylakoid, the granum. So we will discuss in the next chapter. Electron transport chain. So you look at this picture. This is a membrane. So these are the electron transport chain. We call it as ETC. ETC. Have to establish proton concentration gradient. What does it mean? When you look at this word, proton concentration gradient, hydrogen ion moving from high concentration to low concentration. Okay, so by looking at this picture, right, use red color. So moving from A to B or B to A is from high to low. Okay, I repeat again. Talking about the concentration gradient. Concentration gradient is the movement of substances from high to low. Am I right? From the region of high concentration to the region of low concentration. So is A to B passive transport from high to low, or B to A is from high to low? So the answer is B to A. So this part is passive transport, and A to B is active transport. Active, passive. And my next question is, okay, A to B or B to A produce ATP. Okay, so we will know the answer in part 3. Okay, so continue. Electron transport chain helps to establish the proton concentration gradient. This ETC establish proton concentration gradient. So means what? Means at the beginning is balanced, but this ETC helping up to make one place with high concentration, so that another place compare relatively is lower. So you look at this picture. You see there is a pump here. Okay, moving hydrogen from the inside, I mean the matrix, to the intercellular spaces. Okay, so at the beginning is balanced. So when pumping out from the matrix into the intercellular spaces, I mean the intermembrane space, sorry, mentioned wrongly. So the concentration of the hydrogen ion here will be higher. So it forms a proton concentration gradient. Okay, continue. high electron and energy electron move from carrier to carrier, from one carrier to another, okay, and releasing energy. Energy is used to pump the proton across inner membrane into the intermembrane space. So when the electron is being moved, okay, from one protein to another protein, I call it as protein, okay, carrier to carrier, so energy is being released here, and the energy release is used to pump the hydrogen ion from A to B, okay. The protons are pumped from low concentration in a matrix to a higher concentration in the intermembrane space. Okay, so the proton move from down the concentration gradient into the matrix. So after that, the hydrogen ion will be so-called diffused from high to low concentration from the intermembrane space back to the matrix, and the energy is released. So when moving back to the matrix, you look at this part, ADP join with the phosphate, phosphorylation occur, ATP is being produced. Proton moves through ATP synthase, this blue color protein or enzyme is ATP synthase to drive the phosphorylation of the ATP. So phosphate joined with the ADP is called phosphorylation. And you see oxygen is being involved. You still remember respiration, glucose plus what? Glucose plus oxygen. Okay, so oxygen is involved now. Okay, act as a final electron and a proton acceptor. So this oxygen accepts electron to form water. Okay, and most of the ATP made through this way. So producing 32 to 34 ATP per glucose molecule. So probably you are wondering, okay, now I know oxygen is involved here, but what about glucose? So we haven't started to discuss about that part, which is in part 3. So comparison between these two phosphorylation, one is substrate link phosphorylation, another one is chemiosmosis. Both are phosphorylation, involving phosphorylation, ATP synthesis. Okay, so substrate link phosphorylation, phosphate from a substrate, any substrate, which is phosphate rich. Okay, substrate is directly transferred to ATP to form ADP to form ATP. Use energy directly provided by another chemical reaction. So the energy needed to form ATP, and the energy is obtained from another chemical reaction. Chemiosmosis, the energy released by the movement of hydrogen ion down the concentration gradient, passive transport, okay, to synthesize ATP. Oxygen act as a final hydrogen acceptor. Location for substrate link, cytoplasm or the matrix. Chemiosmosis happen in the inner mitochondrial membrane and also the thylakoid membrane of the chloroplast. The quantity of ATP for the substrate link is small, but the chemiosmosis is large.